Good evening and welcome to the week 10 live session of experimental biochemistry. I am Surasree. I am a Prime Minister's Research Fellow working in the Department of Biotechnology in IIT Kharagpur. In today's session, we are going to talk about very preliminary, very preliminary introduction about recombinant DNA technology. So, in the previous session, we were talking about the technique for DNA isolation, so plasmid DNA isolation as well as genomic DNA isolation. And we also learned the structure of DNA, the structure of RNA, the difference between DNA and RNA, and the method of isolating plasmid DNA as well as genomic DNA. Now, in today's session, we are going to cover these topics, recombinant DNA technology, overview of gene cloning, some tools which are required for genetic engineering, as well as PCR or polymerase chain reaction. Now, what is recombinant DNA technology? So, recombinant DNA technology is manipulation of an organism's genome, and this recombinant DNA technology can involve various changes. So these changes are done on the DNA. These changes can be mutation in the single base, in a single base. It can be um, deletion of a certain stretch of um, gene, or it can be insertion of a stretch of a gene or a stretch of a DNA sequence. Now, single base mutation means that it is a change in one single nucleotide of the DNA, whereas deletion, insertion, these involve a certain stretch of the nucleic acid sequence. So either you can delete it or we can insert a foreign stretch of sequence in one particular DNA. So these are the changes that we try to manipulate on an organism's genome. And for that, what we do is we extract DNA from one organism's genome and we combine it with the DNA of another organism. And that is the principle of recombinant DNA technology. So it is recombination between the genome of two organisms, and this involves primarily changes that we make on our nucleic acid, which is DNA. Now there are some components of the recombinant DNA technology. Number one is the gene of interest. So gene of interest means the DNA segment which we want to manipulate, which we want to change, which we want to either it can be a single base change or change of a certain stretch of a nucleic acid. So that is actually the gene of interest. Next, coming to plasmid. So plasmid, we were talking. One minute, my screen just froze. Plasmid is in the previous session we also discussed about plasmid DNA. It is circular double-stranded extra chromosomal DNA which can be used as a vector. So what is a vector? A vector is a carrier DNA molecule. So it is a DNA molecule to carry any foreign genetic material in the host. So we suppose we want to make some changes, and for that we make the changes or insert the gene into the plasmid, and we use that plasmid as a vector to carry some foreign genetic material. And then what we do is we transform that DNA molecule into the uh, host cell. So it is the transfer of the gene of interest into the host cell. And then what we do is that we make clones of that organism. So clones is the exact copies. So clones are the organisms which carry identical genes. So on these terms, I'm going to elaborate later on. But first, let's talk about an overview of gene cloning. Okay. So, what is gene cloning? So let's talk about it step by step. So what we first do is that we isolate the plasmid from a bacterial strain. Okay. Then what we do is we manipulate the plasmid according to our own interest, right? So then what we do is we manipulate the plasmid. So we cut the plasmid. We cut the plasmid with enzyme, which enzyme I'm going to discuss later on. Then what we do is we combine this plasmid with our gene of interest. So this gene of interest, where do we get? So this is plasmid isolation. This is genomic DNA isolation. Okay, the genomic DNA is isolated here. And this particular DNA, this particular red stretch of DNA, okay, this is also cut with the same enzymes as that of the plasmid. Right? So the plasmid and this particular stretch of DNA has to be cut with the same enzymes, and then such that they can be joined together. So this particular stretch of DNA can now be joined into this plasmid vector. So the first step is the isolation of plasmid and isolation of the DNA that we want to manipulate. Next, we cut the plasmid as well as the DNA with the same enzymes. Then the DNA portion which is cut and the plasmid which is cut, these two are combined with an enzyme which is called DNA ligase. This is the process of ligation, and then what we do is that we put this ligated plasmid. So this plasmid is now again circular. Okay, this plasmid, when it was cut, it became linear, but when it is joined again with ligase, it has become circular again. Now this circular plasmid is transferred to a bacterial host. This is called transformation. Okay, so now we are transferring this plasmid into the bacteria. The bacteria will uptake the DNA, and then the bacteria will make copies of it. So the interesting point about plasmids is that they can replicate on their own. So obviously this plasmid will replicate inside the bacterial cell, and when the bacteria will grow, the number of plasmid will also grow. So this process is transformation, right? So this is the initial part of this gene cloning, that the plasmid DNA is cut and it is joined with the DNA of interest, and then this ligated plasmid, this joined plasmid is transformed into host cell, and then these bacteria will be grown to produce these colonies. So from these colonies, we will pick one colony and then we will put it into large culture. Then we will, what we will do, we will use an inducer molecule to induce our gene of interest, so that from the plasmid DNA, the mRNA of my gene of interest will be produced, and also the protein will be produced. Right. So this is the recombinant plasmid. I am going to discuss about the features of the recombinant plasmid. But this plasmid is transformed into this bacterial cell. Then this bacteria can be grown on an antibiotic plate, because these recombinant plasmid, they have an antibiotic resistance gene. So I'm going to discuss about these things in much more detail. But this is the overview that how gene cloning can be done. Is there any doubt? Okay, if there is no doubt, let's proceed to the tools of genetic engineering. So this part that I was talking about, the cutting of the plasmid. Okay, so this cutting of the plasmid DNA as well as any DNA of our interest, how do we do that? We use an enzyme which is called the restriction endonuclease. Now, these are enzymes which are endonuclease. So, they can cleave the cleave between two nucleotides. So they can break the phosphodiester bond between two nucleotides in the middle of the DNA sequence. So suppose this is my DNA sequence, and the restriction endonuclease can make cuts on the DNA sequence like that, inside the DNA sequence. That is why it is called endonuclease, right? And the important feature of such restriction endonucleases is that they can recognize a particular stretch or a particular set of nucleotides. So each restriction endonuclease has its unique sequence of recognition. Okay, so that is called a restriction site. So if that set of nucleotides is present anywhere in the DNA, the restriction endonuclease will be able to cut at that particular sequence of DNA. Okay, so for example, here you can see that the top row of this figure, you can see that Hind3 digest is there. Hind3 digest. So Hind3 is a restriction endonuclease. So these restriction endonucleases are named after the bacterial strain from which it is isolated. Okay. So for example, ECO R1. ECO R1 is a restriction endonuclease, and the ECO R1 has received its name from the bacteria Escherichia coli, strain R. So E stands for Escherichia, Co stands for coli, R stands for the strain R, and I, or ECOR1 actually, EQR1 stands for the first enzyme that was isolated from E. coli, right? So that is how the names of the restriction endonucleases are given. These restriction endonucleases are naturally present in the bacterial system, right? In the bacterial cells, these are naturally present, and from those bacterial cells, these enzymes can be isolated. 1 minute, I'm having this network issue, not being able to share my screen. Just give me a moment, please. Am I audible and visible? Okay. All right. So I was discussing about these restriction endonucleases. So they have a particular restriction site. Now, this restriction site, the feature of this restriction site is that these restriction sites are usually palindromic. So, which means when you read them from both ends, they will be the same sequence. So, from this 5' to 3' end, it is AAGCTT. From the again in the reverse direction, in the complementary strand, it is AAGCTT. Right? So, this is why it is called a palindromic sequence. So, when from whichever side you read it, it will be the same sequence. So these kind of palindromic sequences are usually recognized by these restriction endonucleases. Now, these sequences that the restriction endonuclease can recognize, this sequence can be a 6 base length or 8 base length, or even its length can be 4 bases also. So whether it is a 4 base, um, whether it recognizes a sequence which is a 4 base long sequence, then it is called a 4 cutter. Then if it recognizes 6 uh, base long sequence, it is called a hexacutter. And if it recognizes a sequence which is 8 bases long, it is called an octacutter, like that. So restriction sites can be either 4 bases, 6 bases, or 8 bases long. And these restriction endonucleases are naturally available in bacteria, and their names suggest from which bacteria they are isolated. So this EcoR5 is the fifth enzyme that is isolated from E. coli and strain R. Okay, and EcoR1 is the first enzyme which is isolated from E. coli or Escherichia coli strain R. So now one other important feature of these restriction endonucleases is that they can recognize these sequences, and they can cut these sequences at a particular site. Now, based on the region where they can cut the cut the DNA sequence, these restriction endonucleases can produce either blunt ends or sticky ends. Blunt ends or sticky ends. What are blunt ends? So blunt ends are basically the ends where there is no overhang. So for example, this type of cut site where the enzyme can recognize this particular sequence and cut it from the middle. So when the enzyme can cut it from the middle, then it produces these blunt ends. But when the enzyme cuts the sequence from like this, for example, after one base, after one nucleotide, right? Here, in case of HIMS3, in case of PST1, then it produces the sticky ends. So these ends are called this type of ends where there is some protrusion, where there are some overhangs. These are called the overhangs. These overhangs are these overhangs can be generated by the endonuclease action of these restriction enzymes, and these are called sticky ends because the ends that are produced by the action of the same enzyme, they can be ligated together, they can stick to each other. So by sticking, I mean to say they are complementary to each other, so they can form base pairs, right? So that is why these ends are called sticky ends. So that is the primary discussion about the restriction endonucleases. Now, the next tool that we are going to discuss is the is another enzyme which is called DNA ligase. So DNA ligase is the enzyme which is responsible for ligating two ends of DNA. So for example, after these restriction enzymes, so what we do with the restriction enzymes? So we cut the plasmid at a particular site with these restriction enzymes. Now, these restriction sites, which are unique for the restriction enzymes, if these restriction sites are present in the plasmid, then we will be able to cut the plasmid with that particular restriction enzyme. So that is what is actually done. So the plasmid is digested with the restriction enzyme, and after that, the the DNA of interest, the gene of interest is also digested with the restriction enzyme. And it should be kept in mind that they are digested with the same enzymes. So, for example, so suppose our this is our plasmid, it's a double-stranded circular molecule. This plasmid is digested with two enzymes, ND1 and BAMH1, and their restriction sites are placed like this, here and here. So, of course, when we will digest the plasmid DNA with these two enzymes, ND1 and BAMH1, then this internal portion, this particular internal portion will be chopped off, right? And this will form a linear DNA, right? Because the circular plasmid will not be circular anymore because it's digestion, so it will be linear, right? Now, there is our DNA of interest. Suppose there is our DNA of interest, right? And this DNA of interest, or if it has to be inserted in between, then this DNA of interest also has to be cut with these particular restriction enzymes ND1 and BAMH1. So this has to be maintained that the plasmid DNA as well as the DNA of interest, they will have to be digested by the same restriction enzymes. And then, so what is the benefit of that? This particular DNA molecule will also be linear, and it will produce the same kind of sticky ends. The same kind of sticky ends will be produced on the two sides of the linear DNA molecule. Now, these sticky ends can be joined together by this enzyme, by this particular enzyme, DNA ligase. Right? Now, this ligase enzyme, this is the crystal structure of DNA ligase from bacteriophage T7. So, from different bacteriophages, these DNA ligases are isolated. For example, T4 DNA ligase. It is very commonly used, T4 DNA ligase. Now, what it does is that it joins the two sticky ends. So, here in this diagram, you can see these kind of sticky ends are formed by the action of restriction endonuclease. These sticky ends can be joined by this enzyme. So, this is the enzyme molecule. This is the ligase which can join these two sticky ends. And in this reaction, it requires ATP. So it is an ATP dependent DNA ligase. So in this reaction, the ATP molecule will be required. So the energy of hydrolysis of ATP is required for such joining of two DNA molecules. So this is the function of the ligase enzyme. Now let's talk about the plasmid vector. So this plasmid vector, there are several important features of this plasmid vector. For the first one is that it is self-replicating. So the plasmid vector is known to have a sequence which is called origin of replication. So what is origin of replication, or it is known as the ORI sequence? So it is the sequence where replication of this plasmid vector can begin. So here this is the sequence. This is the map of a plasmid. This is the map of a particular recombinant plasmid. So this plasmid is not naturally available in bacteria, but this is generated in lab. manipulated in lab for some particular experiments. And this is the map of such a plasmid. If you see in details, so this particular yellow sequence that is shown here, this is the origin of replication. This is the ORI sequence where the replication of the plasmid can begin. And it is self-replicating, so its replication does not depend on the replication of the genomic DNA of the bacteria. So it can replicate on its own without the help from the genomic DNA of the bacteria. So this is the ORI sequence. Now the next important feature of plasmid vector is the multiple cloning site, which is also known as the MCS. Now the MCS is actually why is it called multiple cloning site? Because this site contains unique restriction sequences. So this site is generated such that it contains unique restriction sequences which cannot be found elsewhere in the plasmid, right? So here we can see that this particular site, for example, you can see this is the MCS region where these are the enzymes, these are the restriction enzymes whose sites are present, like HIM3, SAC1, EcoR1, XBA1, SPH1, right? So these are several restriction endonucleases whose sites are present, whose restriction sites are present in this multiple cloning site, right? And the importance of these sites is that these are unique, so they are present only here in this entire plasmid DNA and not elsewhere. Why is it important? So that when you digest the plasmid DNA with a particular restriction enzyme, it only cuts at one site, at one unique site, and not elsewhere in the plasmid. So that has to be maintained so that your plasmid is not chopped off at many places. Otherwise, your purpose will not be fulfilled. So that is why this multiple cloning site is very important, which contains these unique restriction sequences. Next, coming to the selectable marker. So this selectable marker is something which selects the plasmid, which selects the propagation of plasmid in the host strain, in the host bacterial strain. So this selectable marker is usually an antibiotic resistance gene. It can also be markers like fluorescent proteins, like for example GFP, green fluorescent protein, RFP, red fluorescent protein, or it can be these antibiotic resistance genes. So here we can see in this map, this is the selectable marker. This is the selectable marker, which is the gene which gives resistance to the kanamycin antibiotic. So CAN-R is basically resistance. Yes, I'm discussing that. So CAN-R is basically resistance to kanamycin sequence. So why is it resistance? Why is this resistance important? So that when we grow these plasmid vectors inside bacterial strain, so then what happens? Uh, the the plasmid, which the plasmid will not be present uniformly in all bacterial cells, right? So we need to select between the cells which bear the plasmid and the cells which does not bear the plasmid. To select that kind of cells which bear the plasmid, we put this antibiotic pressure so that the cells which do not have the plasmid cannot produce any resistance against this antibiotic kanamycin. So of course they will die in the presence of this antibiotic. The antibiotic will kill those cells, but the plasmid is but the cells which will have the plasmid, they will always survive in this antibiotic resistance, right? despite this antibiotic resistance. So that is why we put the selectable marker to select the plasmid vector and to select the cells which possess the plasmid vector, and as well as we also maintain the propagation of this plasmid vector, so that if so these plasmids are, they are not essential components of the bacteria. Okay, so they are extra chromosomal, so the function of the bacteria will not entirely depend on this plasmid vector. So the plasmid vector can be lost also in between in, in several generations of the bacteria. So to prevent that, to prevent loss of the plasmid vector, we um, put an antibiotic pressure so that the plasmid vector cannot be lost during the process, during several generations of the bacterial cell, right? Next, the next point is the promoter. The promoter sequence is, obviously, you know that if you have studied transcription, so the promoter sequence is important for the transcription of our gene of interest, right? Now, there can be several promoters that will be present in the plasmid. So, for example, this is T7 promoter. T7 is actually the T7 bacteriophage. So, this promoter sequence is taken from T7 bacteriophage. Why T7 bacteriophage promoter sequence can be taken? Why is it important to take the T7 bacteriophage promoter? Any idea? Okay, so basically this T7 promoter, actually this is the promoter for the T7 RNA polymerase gene of the bacteriophage T7. Okay, so this promoter is associated with the production of RNA polymerase gene of the bacteriophage T7. Now, this RNA polymerase, as you must understand, that bacteriophage is the virus, so it needs to produce a huge amount of mRNA and proteins. So it's T7 RNA polymerase. The polymerase will be produced in much large amount, higher amount, and the promoters can actually be strong promoters or weak promoters. Sorry. The promoters can be strong promoters or weak promoters. Strong promoters means promoters from which the rate of gene expression is much higher. So it is responsible for high amount of gene expression. Whereas the weak promoters are the promoters where the association between the promoter and the RNA polymerase is less. So that is why they are weak promoter, and the gene expression is less from those kind of promoters. So this T7 promoter is a strong promoter from which there can be a huge amount of gene expression. So which is why this promoter sequence is placed in close proximity to the MCS, the multiple cloning site, so that our gene of interest can be inserted after this T7 promoter sequence, right? So the our gene of interest can be expressed by this T7 promoter sequence, right? Which is why this T7 promoter is present here, the T7 RNA polymerase promoter from the T7 bacteriophage. Okay, now you can see here that the plasmid also is of small size. So we discussed this in our previous session, that the DNA, the difference between plasmid DNA and genomic DNA is that genomic DNA is much larger in size, but the plasmid DNA approximately ranges from 5 to 10 kilobases. So that is in the range of kilobases, right? So Plasmid DNA is much smaller in size, so it is easy to handle. It is easy to manipulate and isolate and uh, perform this restriction digestion and so on. So that is why plasmid DNA is preferred as vector. And the point that you were asking, that is the F1 origin. The F1 origin is actually a phage derived origin. Again, it is derived from a phage uh, that allows replication and packaging of the single strand DNA into phage particles. So this F1 origin is derived from phage, which allows this plasmid to replicate in phage and to produce these phage particles, right? So that is the function of the several components of a plasmid DNA. The kanamycin resistance gene or any such antibiotic resistance gene can be produced, can be present, like for example, ampicillin resistance gene. So these are selectable markers. There is an origin of replication. There is a multiple cloning site where the restriction enzyme sequences are present, where the gene of interest can be inserted, can be cloned, and it is under a promoter, which has to be a strong promoter for better expression of our gene of interest, right? So these are the components of a plasmid vector where we are going to insert our gene of interest, right? And then once we insert our gene of interest in the plasmid, then the next thing that we are going to do is that we need to transform this plasmid in a host strain. So this transformation has to be done in a strain which can produce our gene of interest in multiple copies, and also it can produce uh, the required protein from this gene of interest, right? So if we consider that protein production is the ultimate goal of such cloning process, then there can be multiple hosts which can be used for this transformation. So it can be bacteria, it can be yeasts, it can be plant cells or mammalian cells. Now the difference between such hosts is that, first of all, bacteria is a prokaryotic system. So bacteria is prokaryotic, whereas yeast, sorry, yeast, plant cells, and mammalian cells, all of these are actually eukaryotic cells. Now, the difference between prokaryotic and eukaryotic system is that the post-translational modifications which occur in eukaryotic system does not occur in prokaryotes. So, after production of proteins, so when after the proteins are produced, in eukaryotic system, there are several post-translational modifications that are performed on the proteins. For example, it can be phosphorylation, it can be acetylation. So these kind of post-translational modifications are not done, are not observed in prokaryotic systems. So if we want to express any eukaryotic protein in a prokaryotic system, then we have to be careful, because these post-translational modifications will not occur in such prokaryotic system, and which is why if our protein, suppose it is not functional without such post-translational modification, then after we isolate the protein from such prokaryotic system, it might not remain functional because of the absence of post-translational modification. But the advantage of using prokaryotic system is that it has very less doubling time. So we know the doubling time of bacteria is approximately 20 minutes. So in 20 minutes, one generation is completed. So of course, if we want to produce a particular protein in bacteria, then it will be very, it will produce very quickly in bacteria. But the problem in such system is that there is a codon bias which exists between prokaryotes and eukaryotes. So what is codon bias? So the codons which are recognized in the prokaryotic system might not be recognized in the eukaryotic system. So suppose, so this comes very crucial when we want to express an eukaryotic protein in a prokaryotic system. So eukaryotic, so what is this problem? If I just elaborate you on that, that is, suppose we have one protein of human, which is, suppose we have human protein X. Okay, we have human protein X with which we want to, so suppose this, we do not know the function of this protein and we want to characterize the function of this protein. So to characterize the function of the protein, what we need to do is we need to purify the protein. For purification of the protein, first we need to overexpress the protein. So for that, what we first do is that we clone this gene of protein X into a plasmid vector. So I'm just going through the process of cloning once more. So what we do is that uh, from human cells, for example, we isolate the DNA and we amplify the gene of interest, the protein X corresponding to the, so I'm writing gene X, which corresponds to the protein X, right? So what we first do is we amplify the gene X. So amplification is done by PCR, which we are going to discuss just after this. So the gene X is amplified by PCR, and then what happens? This PCR product is digested with particular restriction enzymes. And this plasmid, where we want to clone the gene X, this plasmid vector, suppose this is PET, PET28A. So this plasmid vector is also digested with the same restriction enzymes, right? So now we have two digestion products. One is the gene of interest, and another is the plasmid. Now what we do, digested plasmid. So the next thing that we do is we ligate the two. So we mix the two in a particular ratio, the gene of interest and the digested plasmid, and we use the enzyme DNA ligase for ligation of the plasmid with our gene of interest. So now this plasmid will be again double-stranded circular with our gene of interest inserted in between. Suppose this is blue part is our gene of interest, which will now express the protein X, right? So now this plasmid DNA, first of all, what we have to do is that we have to transform it in a bacterial cell. Okay, so this gene of interest is now transformed into a bacterial cell. The plasmid is transformed in a bacterial cell, right? And now this bacteria will make many copies of this gene of interest, right? And if this gene of interest is actually expressed under a inducible promoter, so this is actually the lac promoter. So I hope you are familiar with the lac operon. So lac operon it consists of the promoter, which is the lac promoter, which is inducible by the addition of IPTG. So, or we can also use glucose So glucose is the natural inducer of this lab promoter, and we can use IPTG in our lab. So we use IPTG to induce this lab promoter to produce our to produce mRNA of the gene X, and to finally produce the protein X by translation, right? So finally this protein X can be produced. So finally, this protein X can be produced. Now, this protein X, we need to isolate the protein from the bacterial cells. The several protein purification techniques we had discussed previously, like the nickel NTA, the affinity chromatography, ion exchange chromatography, and so on. Gel exclusion or gel size exclusion chromatography. So by which we will isolate the protein of our interest. So that is the total plan. First we clone the gene, and then we overexpress the gene, then we purify the protein for any functional study of the protein. Now, in this system where this human gene is cloned into the bacterial plasmid and grown in bacterial system, then what may happen is that these protein might have some post-translational modifications which will not be occurring in the bacterial cell when the protein is being produced. Number one. Next thing is the codons, which actually are responsible for bringing certain amino acids, right? The codons specific for certain amino acids, these codons might be different between the bacteria and eukaryotic system. So to prevent such kind of anomaly, what we do is we often um, change the codons to prevent this kind of codon bias, so that suppose there is one amino acid in the protein arginine, for which the codon between the bacterial system and the eukaryotic system is different. So this codon for arginine, we change it to the codon of the bacterial system, so that this amino acid can be produced in the protein of our interest. We need the protein finally. So we need the amino acid, we need the correct amino acids to be introduced in the protein. So that is why we change the codon of our of our protein of interest, that is protein X of human, to the codon of bacteria, such that the amino acid remains unchanged. Right? So that is to prevent the codon bias. Now, for post-translational modification, to avoid absence of post-translational modification, we can also express our protein of interest in eukaryotic system like yeasts. So yeasts are also very easy to grow, and the protein can be collected easily from Saccharomyces cerevisiae. So there are several plasmids which can be grown in Saccharomyces cerevisiae, and our gene of interest can be cloned in such plasmids and grown in yeast. And the other eukaryotic systems, like the plant cells and the mammalian cells, the plant cells and mammalian cells, they can easily express the eukaryotic genes. There will be no such codon bias which will be present. The post-translational modifications can also be carried out, but the difficulty is that mammalian cells, the plant cells, they are not easy to grow, right? And they they have a large doubling time also. So their generation time is much longer. So we need to grow them for long hours, and then we can isolate the protein. Also, the protein production, the yield of the protein will not be very mm, um, large amount. So it is difficult to get large amount of protein from these eukaryotic cells like plant cells and mammalian cells. So naturally, bacteria is the most preferred system of hosts. We can also express our proteins in some insect cells, for example, SF9, Spodoptera frugivorida cells, where our gene of interest can be cloned and transformed. So these are the hosts where we can grow our gene of interest. And finally, overexpress our protein and purify our protein. So next, coming to the part where we will have to amplify our gene of interest. So for that amplification, this process is carried out, which is called polymerase chain reaction. Okay, but before going to that, please let me know if you have any doubts so far in the cloning portion. So these plasmids can be used in which hosts? That is the question, right? So this actually depends on the origin of replication. So if this plasmid contains an origin of replication which can be recognized in the yeast system, then yes, it can be grown in yeast. If the um, origin of replication cannot be recognized in the yeast system, then this plasmid cannot be grown in yeast system. So depending on the origin of replication, so this ORI sequence that I told you, this ORI sequence is recognized by the bacterial system, right? So that is why the the FIT28A plasmid, it can be grown in bacterial system. It can also be grown in phages because it has the F1 origin, right? The phage origin. But if it does not contain any eukaryotic origin of replication, right? So it will require eukaryotic origin of replication to be replicated in yeast. So if it does not have that, it cannot be replicated in yeast, and therefore, if even if we transform it into yeast, it will not be able to replicate and it will be lost. So is that okay? All right. Okay. So next we move on to this polymerase chain reaction, which is commonly called PCR. So PCR was first discovered by Karamolis, scientist Karamolis, and he had received Nobel Prize for the discovery of polymerase chain reaction. So this polymerase chain reaction is usually done in a machine which is called a thermocycler machine. So what is it? So what is it? It is basically a cycle. It actually can cycle between temperatures. It can change. There can be changes of temperature in this machine, and by that, several parts of the reaction can be carried out by maintaining different temperature. So, what are the different parts of this PCR reaction or polymerase chain reaction? First of all, the components of the PCR reaction will require a DNA template, so which needs to be amplified, right? So there has to be a DNA template. Now, this DNA template uh, can be amplified by the presence of these primers. So these are called the DNA primers. So these are the DNA primers. Okay. So what is the function of these DNA primers? These DNA primers are complementary to one part of the DNA template. It can sit on the DNA template because it is complementary, and then it provides a free 3' hydroxyl end from where the replication can be carried out, right? Now, one essential part of this process where the DNA will be amplified is replication. For this replication, there has to be a polymerase. Now, this polymerase is actually a thermostable polymerase. Why is it a thermostable polymerase? Why such a polymerase is required? We will discuss, because this reaction occurs at different temperatures. So in the first step of this reaction, there has to be a denaturation. Denaturation means the two strands of, these are the two strands of the DNA template. These two strands has to be first separated from each other. So this is the process of denaturation, where the two strands of the template are separated from each other, right? Or denatured. They are made single strand. Why are they made single strand? So that now the primers can anneal, the primers can anneal to the single strand template DNA, right? That is called annealing of primers. Now this denaturation is done at a higher temperature like 95 degrees centigrade, right? So when we start the reaction, the first step that we do is denaturation. And when we denature the DNA, the polymerase is present in the reaction. So at 95 degrees centigrade, the denaturation is occurring, and the polymerase has to resist such a temperature, right? And the polymerase cannot be degraded in such a temperature, because we know that a polymerase is a protein, and we have studied this before, that proteins are degraded at such high temperature, right? Which is why we boil the proteins at such a temperature and then run it on SDS page, right? So proteins are usually degraded at such temperature. So which is why we require a thermostable polymerase, which will not be degraded at this temperature, right? So this thermostable polymerase is isolated from a bacteria called Thermus aquaticus. So this, from the name of this bacteria, this polymerase is called Taq polymerase, which is a thermostable polymerase. So now, in the first step, the denaturation has occurred, and the two DNA strands are separated from each other. And then primers, which are complementary to the DNA strand, can sit on the template strand. And then the next thing that will happen is elongation or extension. We can say extension or elongation. So now the amplification will begin. So the replication will begin. So the polymerase will begin its function. So it will polymerize the DNA, the complementary strand of the DNA from this primer. So this is the primer from which provides a 3' hydroxyl end, from which end the polymerase starts elongation. The polymerase starts elongating the complementary strand to this template strand, right? This occurs for both the strands, right? So in this elongation step, the polymerase will be able to copy the DNA, and then what will happen? Then this particular steps will be again repeated. So it will occur in a cycle. Okay. So in the first cycle, after this denaturation, annealing, and extension, after the first cycle, from one, so for if there is one DNA molecule in the beginning, if there is one DNA molecule in the beginning, after the first cycle, two DNA molecules will be produced, 1 and 2, right? Two DNA molecules will be produced after the first cycle. Then after the second cycle, from two DNA molecules, four DNA molecules can be produced. 1, 2, 3, 4, right? So here the number of copies are increasing exponentially, right? So that is why it's called an exponential amplification. And we will carry out this reaction for approximately 25 to 30 cycles. Okay, so in this cycle, first, in each of the cycles, first the temperature will rise to 95 degrees centigrade, denaturation will occur, then the temperature is again brought down so that at this less temperature, the primers can anneal to the strand of DNA. Then again, a temperature is maintained in which the polymerase can elongate. So this elongation temperature is dependent on the polymerase action. Then again, after that, when the elongation is complete and the DNA strands are formed, then again the cycle goes back, and it begins with denaturation again. So again the temperature is increased, denaturation starts. Right, so this is carried out in a cycle, and that is why it's called a chain reaction, polymerase chain reaction, right? And it is performed in this machine, which is called a thermocycler. So now let's talk about the components which are required for this PCR reaction. So of course, the first component is the DNA sample, the template DNA, which we need to amplify. Next are the DNA primers. So these are short stretches of DNA which are complementary to the template DNA. Why are they essential? They are essential for providing a 3' hydroxyl end from where the polymerase can start elongation. Next are the nucleotides. So now, once the polymerase is elongating, and since this is occurring outside the cell, right? This is not an in vivo activity, right? So this is occurring inside a tube. So we need to we need to supply the nucleotides with which the polymerase will be able to elongate or amplify the DNA, right? So these nucleotides, or which are called the DNTPs, deoxynucleotide triphosphates, right? So these will have four nucleotides: DATP, DGTP, DCTP, and DTTP. So adenine, guanine, cytosine, and thymine, right? So will be four DNTPs which will be mixed in equal ratio, and it needs to be supplied in this PCR reaction. Next, the Taq polymerase, the thermostable polymerase, and there should be a buffer in which the reaction is carried out. Why is this buffer required? Because there is an enzyme which which will elongate the DNA molecule, and for the function of this enzyme, we need to provide a certain buffer in which the enzyme will be most functional. So this buffer is provided, right? And in this buffer, several components are there. For example, magnesium ions. This is also present in the buffer, and this total reaction is carried out in this small tube, which is called a PCR tube. So in a PCR tube, we mix these components, and in the end, we add the PCR enzyme, which is the Taq polymerase, and then we put the PCR tube in such a thermal cycler machine, and we set the cycle. So this cycle also is very important. So this cycle has to be set so that denaturation, annealing, extension can be done cyclically, and then we allow the thermal cycler to amplify. So we put the reaction in this thermal cycler so that the thermal cycler can maintain the temperature, and then the polymerase will perform its function, right? So in the polymerase chain reaction, so this is also again showing you the one cycle and the step-by-step process of this, that in the first step there is denaturation. So the two strands, strand A and strand B, these are denatured, these are made denatured in a high temperature. When the denaturation occurs, then there is annealing of the, we reduce the temperature to approximately 50 to 60 degrees centigrade. And in this temperature, the two primers, so these are the primers, short stretches of DNA which are complementary to the template DNA. This can anneal to the template DNA. And then finally, this is the 3' hydroxyl end of the primer from which the elongation begins. So this elongation or extension occurs at a temperature which is the optimum temperature for the enzyme Taq polymerase. So approximately it is 72 degrees centigrade. So this is how the PCR reaction occurs. Now, talking about these primers. So these primers are sequences which need to be designed by us such that this primer can function in a way such that it can anneal to the template DNA. So it has to be complementary to the template DNA. It has to be um, designed in such a way that it is complementary to the template DNA. And there has to be two primers actually. There has to be two primers. So the DNA strands, the two DNA strands, they are 5' to 3' and 3' to 5', right? So they are anti-parallel to each other. Now the primers, there will be two primers. Forward primer, there is one forward primer and there is one reverse primer. Forward primer and reverse primer. So the difference between forward and reverse primer is that they are complementary to two opposite strands of the DNA. The forward primer is actually complementary to 3' to 5' strand of this DNA. So this forward primer, which has these ends, 5' and 3', right? So this forward primer will sit on this particular strand, and from this 3' hydroxyl end, the elongation will begin in this direction. Okay, so this is the function of the forward primer. Now the reverse primer will sit on the opposite strand. So suppose this black one is the reverse primer. So the reverse primer will be annealed here. This is the reverse primer. So this reverse primer will have 5' and 3' end, and this end will have the free 3' hydroxyl, which will again help in elongation by the polymerase. Okay, so there are two primers, forward primer and reverse primer, and they will sit on the DNA like this, on the DNA template like this. And these primers, the forward and reverse primers, they have to be designed in such a way such that they are complementary to the DNA template and they can anneal to the DNA template of the portion which we want to amplify. Now, suppose this is the nucleotide sequence of a particular gene X. This is the nucleotide sequence of a particular gene X, and we need to design two primers to amplify this gene. So the forward primer and the reverse primer we have to design. So of course the forward primer, for the forward primer we will take a certain portion of this gene and that will actually serve as the forward primer. So this primer, so this one is the forward primer. This one is the sequence of the forward primer. Now there has to be, the forward primer not only has to be complementary to the DNA template, but it also has to contain a particular sequence which will be the restriction site, which will be the restriction site. So this is the restriction site for ND1 enzyme, which is why I have named the primer with this forward ND1, and the reverse primer contains the damage 1 restriction site. So these sites can be recognized by the enzymes ND1 and damage 1, right? And they can be digested from this place. So why do we provide this kind of restriction sites along with the primer? So that when our DNA is amplified with these restriction sequences, right? And we can cut the DNA with these from these restriction sites and then we can ligate the DNA with our plasmid vector. Okay, so for that purpose, we will provide these restriction sites in the plasmid, and these restriction sites will be cloned, or sorry, the restriction sites will be inserted towards the 5' end of the forward and the reverse primer. So the second one is the reverse primer. First one is the forward primer. Now, if we go from 5' to 3' end, the 5' part of the gene, uh, so you can see it from here. This part, this part of the forward primer after the restriction site is actually same with the first portion of the gene. So you can see it here: ATG, ATG, GCC, GCC, AGC, AGC, AGA, AGA, GG. So this is exactly the same sequence of the 5' end of the gene, right? And 5' to this sequence, we have kept this restriction site sequence, and before that, this is actually an overhang. Why is this overhang kept? So that it actually helps in positioning of the restriction endonuclease. The restriction endonuclease can be positioned um, on the overhang. So the overhang is maintained for that purpose. And after the overhang, we have kept, so here, this is a long overhang. And here, in this case, you can see that for damaged one restriction site, we have put a small overhang of 2 nucleotides only. So after the overhang, we have kept the restriction site, the ND1 restriction site for the forward primer, and the BAMH1 restriction site for the reverse primer. Then in the forward primer, we have kept the 5' sequence of the gene. Now, as you must understand that this particular forward primer will not be complementary to this sequence because it is the same sequence. So there will be no complementarity, but this exact same sequence will be complementary to the reverse, to the opposite strand, which is the 3' to 5' strand, this particular strand. So if the forward primer has a sequence of this part, it will obviously not be able to anneal to this strand, but it will be able to anneal to this particular strand, right? Because it is complementary sequence to the 3' to 5' strand. So that is why the forward primer will anneal to the 3' to 5' strand. Whereas the reverse primer, actually will have this 3' end sequence. But in reverse and in reverse complementary sequence. So after the overhang and the restriction site sequence, we will put the reverse complementary part sequence of this region. Why reverse? Because this is 5', this is written in 5' to 3'. So this particular reverse primer will sit on the 5' to 3' strand, right? So that is why it is reversed. And then it is complementary to this particular sequence because this is the sequence of the 5' to 3' strand. This is the sequence of the 5' to 3' strand. So we need to design a primer which is reverse to this and complementary to this, right? So which is why we have reversed and complementary the sequence of this. So you can see here it is TGA or AGT, you can see from this particular side, AGT. So here we have kept complementary sequence of that. So So we can see A is complementary to T, G is complementary to C, T is complementary to A, C is complementary to G, T is complementary to A, T is complementary to A, like that, right? So the complementary and reverse sequence is used for the reverse primer. So this way the primers are designed so that they can be complementary to the two strands, the 5' to 3' strand and 3' to 5' strands of the DNA template. Is that okay so far? All right. So for cloning of the gene, I told you that the genetic code also has to be kept in mind. So this is the codon chart, right? So you will see the codons and the respective amino acids. Like for example, UUU, UUC, these are coded by phenylalanine. UUA, UUG, these are codons for leucine, right? So these are the codons. And we can see here some important codons, which is AUG for methionine. So this is a single codon amino acid. And these are the stop codons, the UAA, UAG, and UGA. Right? These are the stop codons, which means when these codons will be present in the mRNA sequence, they will not be recognized by any amino acid, but they will be a stop signal. So there, the protein synthesis will stop when these codons will be reached. So this is the genetic code, and the genetic code also has to be taken care of when the eukaryotic proteins are being expressed in prokaryotic system and so on. Next, coming to the PCR reaction, how the reaction will look like. So I told you that it is carried in cycle. So the first step which is carried out, so this is the temperature and this is the time. In this manner, the PCR reaction scheme is actually shown. This is the temperature, and below that line there is this time. So the first step is initial denaturation, where we allow 2 minutes of time to initially denature all the DNA template. And then this particular part goes in cycle. So this is the cyclical part. And this is carried out in cycle. And this 30x actually means that there will be 30 cycles. We can also do it for 25 cycles or 35 cycles, so on, right? Now this in this cycle there are three steps. This one is the denaturation step. Then this one is the annealing step, where the primers will anneal to the DNA template. And the third one is the extension or elongation step. This extension temperature is based on the optimum temperature for the polymerase. And this extension time is also based on the rate of polymerization of the polymerase. So extension rate, for example, Taq polymerase, one particular Taq polymerase has a extension rate of 1 kb per minute. 1 kb per minute means that in 1 minute it can amplify 1 kilobases of nucleotide sequence, right? So suppose our DNA is 0.5 kilobases in length. The DNA that we want to amplify, it is 0.5 kilobases in length. Then we will provide 30 seconds time for this extension to happen. So this is why we have kept 30 seconds here. Suppose our DNA is 3 kilobases in length, then we will keep 3 minutes time in this particular extension step. Now this will be carried out for 30 cycles, so that at the end of 30 cycles, exponentially the number of DNA molecules can be produced. So in one cycle, 2 molecules of DNA are produced. In 2 cycles, 4 molecules of DNA are produced. In 3 cycles, after 3 cycles, 8 molecules of DNA are produced. So it is basically 2 to the power n. So it is 2 to the power 2, it is 2 to the power 3. So n is the number of cycles. So if n is the number of cycles, the amount of DNA which will be, or the molecules of DNA which will be produced after n cycles will be equal to 2 to the power n. Right? So it is basically an exponential amplification. So this is carried out in cycles, and then finally the last step will be final elongation or final extension step, after which the temperature will be brought down to 4 degrees centigrade, and the PCR reaction can be stored or can be kept in 4 degrees centigrade for hold. So that is the end of the reaction. So begins with initial denaturation, then cyclically denaturation, annealing and extension, and followed by final extension where we allow some time for the polymerase to extend any leftover product which might be there. And then finally this reaction is stopped by taking down the temperature to 4 degrees centigrade. So this is the PCR reaction scheme. And now these are some shell images. So this is a 1% agarose gel. This is the DNA ladder you can see here, right? Now the PCR product, I told you that it is a 0.5 kilobases in length, so which is 500 base pair. So uh, it's faintly visible here. So this band is 250 base pair, and the upper band is 500 base pair. And when we run the PCR product on 1% agarose. So after this PCR amplification, we run the PCR product on 1% agarose gel, and we can see these two bands. So these are my PCR product, and they have the same size as that of the 500 base pair marker. So we know that our PCR product is 500 base pair in length, and therefore there has been no non-specific bands which are present here. So there is no other band which is present in this PCR product, right? So that means the reaction has been specific, right? So that is the PCR product, and then we can digest our PCR product, right? So we can digest the PCR product, and then we can ligate the PCR product with the plasmid, and then we can transform it in the bacteria, and then we isolate the plasmid and we confirm whether the plasmid contains our gene of interest or not. For confirming that, we again digest the plasmid with the same restriction enzymes, and here you can see that this is the digested vector, this is the digested insert, which is a 500 base pair in length, and this is actually the clone plasmid. So the clone plasmid contains the plasmid as well as our gene of interest, right? And when we digest this particular plasmid, we can see that this is the vector and this is the insert that has been formed, that has been digested. So this insert is actually inside this clone plasmid, right? Which has formed a band after this digestion process. And okay, so this restriction digestion is then carried out to confirm the presence of this gene of interest in this particular plasmid, and then run on agarose gel. And finally, these plasmids can be transformed into competent cells, and these cells can be either cloning strains or expression strains. So cloning strains, for example, E. coli DH5 alpha. This is a cloning strand. And what is an expression strand? For example, BL21D3 or Rosetta, origami. These are some expression strands. These are all bacterial strands which are actually engineered expression strands. So, what is the role of this BL21D3, for example? So, this is actually the mechanism of action of BL21D3. And what we can observe here is that this D3 is actually derived from lambda D3 fudge. Lambda D3 fudge. From this lambda D3 fudge, this particular sequence is actually um, this particular sequence is combined in the E. coli genome, and so so the point is that this contains a T7 RNA polymerase gene. The portion of this lambda D3 gene, which has been incorporated in the E. coli genome, it contains a portion of the contains the gene of the T7 RNA polymerase. And why is it done? To prevent any leaky expression. So how is that actually performed? So this plasmid DNA, where our gene of interest is cloned, it contains this T7 promoter. Okay, so this T7 promoter can be recognized by the T7 RNA polymerase. Okay, and this T7 RNA polymerase can sit on this T7 promoter and express our gene of interest. So this T7 promoter here is present, the T7 promoter, under which our gene of interest is cloned, can be recognized by T7 RNA polymerase, which is produced by the lysogenic insertion of the D3 gene, the D3 T7 RNA polymerase gene. So this T7 RNA polymerase gene produces this T7 RNA polymerase, which can go and sit on the T7 promoter and express our gene of interest. But this can only be done when there is induction or inducer which is added in the medium, right? So when the leaky, how is that maintained? Now there is another plasmid which is P-lysis. This P-lysis plasmid is actually a plasmid which carries the T7 lysozyme gene. So this lysozyme can degrade this RNA polymerase when it is present in less amount. So when there is leaky expression of this T7 RNA polymerase, that means expression when there is no inducer which is given. Then this lysozyme can can be expressed and from this P-lysis plasmid. So this actually carries this P-lysis extra plasmid which expresses these lysozyme which can cleave the T7 RNA polymerase when it is produced in small amount. But when the inducer is given, so when there is IPTG, when it is added in the medium, it will express large quantities of this T7 RNA polymerase and it will go and sit on the T7 promoter and express our target gene. So that is how it is ensured that our target gene is expressed in large quantities in these expression strains. So Rosetta is such an expression strain where there is P rare. P rare is a plasmid which contains some rare tRNA codons. So rare codons for some tRNAs are present in this Rosetta strain such that the codon bias can be overcome. Origami is a strain where these Um, Post-translational modifications, the sulfide linkages are actually can be formed. So these are some engineered strains which are engineered for optimum expression of our gene of interest, right? So that's all for today. Now let's discuss the question answers. Write your answers here in the chat box, or you can tell me also. Okay, correct. Correct answer D. Next question. Yes, correct answer. The restriction endonucleases. Next question. Yes. So next question. What is this answer? What is the answer of 3? Okay, right. Vector. Correct answer. Next question. Okay. Correct answer is A. Next. Yes. Transformation. Correct. Next. There are two answers here. B is correct. There is another option. Yes, A and B. That is the correct answer. Next, yes, unique restriction site. Next question. Correct answer. Vector will not replicate. Next question. Okay. Polymerase chain reaction is the amplification method. Next. Yes. So D is the correct answer. Next. Yes. Denaturation is the first step in PCR. Next. Yes. Correct answer 5 prime end. Next question. So the stop codon. Okay. So the stop codon is present at the 5 prime end of a reverse prime. So suppose this is the 5 prime to 3 prime strand and this is the 3 prime to 5 prime strand. The reverse primer will sit here, right? This will be the 5 prime to 3 prime end. Now, obviously, if at the 3 prime end stop codon is present, from the 3 prime end this elongation will occur, right? So therefore, the stop codon cannot be present at the 3 prime end of the reverse primer. But the stop codon should be present at the 5 prime end of the reverse primer so that that will stop the uh, translation of the proteins, right? So the stop codon should be present at the 5 prime end of the reverse primer, but at the reverse primer only, not in the forward primer, right? Am I clear? Next question. Okay. Correct answer. And the last question for today. No, D is not the correct answer. What is the difference between expression vector and cloning vector? All cloning vectors will also have selectable marker, origin of replication, but these gene expression control elements, that is only present in the expression vectors, which is why they help in expression of the gene. So the correct answer is B. Okay. All right, so that's it. We end today's session. If you have any doubts, please let me know. And next week, this time, we will again join for the week 11 session. Thank you and all the best.